What can I say about Annie? She's so popular she needed a bigger room. This is not happening, which is strange considering my NMR class barely has 4 people in it. So family and friends to take my NMR class, so we appreciate it. Annie got her Bachelor of Science at the University of Hartford, and she joined my lab in 2013. She was supported between 2015 and 2018 by a very prestigious National Science Foundation Graduate Research Fellowship, which she got in her second try, so that shows she has persistence. That's changed. You can only apply once nowadays, so please write your congressman and tell them to increase our funding. She has 4 papers from my lab, of which she's first author on 3, and 1 is in preparation, and you're not going to get a pass on not working on that after this. Her interest and her research has been on NMR of proteins that have multiple structural states, and she was kind of the go-to person in our lab for NMR structure calculation. Besides her work, she also helped with some other students, including Carol Teschke's students on virus protein structures, and her colleague Becca Newcomer on her protein structure as well. So the title of her talk today is Adventures in Structureland, Exploring Protein Conformational Plasticity by NMR. So please welcome Annie.
Thank you, everyone. I'm really honored to have everyone here, and I've wanted to cry all morning, so hopefully that doesn't happen. Welcome to my talk where I want to teach you about all of the adventures that I've had in Structureland, and I wore the sweatshirt today to kind of be in theme, and we indeed are all mad here. I'm going to dim the lights for this one. All right, so proteins are complex macromolecules that carry out all of life's functions, and they help us in pretty much everything like digestion, movement, sight, and for most of us, brain function. They're made up of amino acids, and they have a 3D structure, or what we call a conformation. These shapes are not static by nature, and they have simultaneous movements that are happening on a range of time scales, ranging from bond vibrations on the femtosecond time scale all the way up to larger domain movements on the order of seconds. Proteins are able to change conformation in order to suit their function. So one example that's shown here is hemoglobin, which is made up of 4 different subunits, which are actually 2 subunits that are put together. When hemoglobin is in high oxygen environments, such as in your lungs, oxygen binds to a heme group that actually changes the subunit interface between the subunits and allows heme to more readily bind oxygen and all of the other subunits. So it gets taken up more readily. When hemoglobin is in a low oxygen environment, so for example, when it goes to your muscles under more acidic conditions, that acidity actually changes the conformation of hemoglobin in a way where it releases the oxygen and the cycle starts again. And this is just one example of how proteins are able to interact with their environment and change conformation in order to carry out a particular function. In other cases, there are intrinsic properties that govern the conformational changes, such as proline isomerization. Amino acids are held together by a peptide bond, and they generally are in the trans configuration, where the side chains of each amino acid are pointing away from each other. The cis configuration is not as favorable because, as you can see, the side chains, when they're on the same side, end up clashing, which is very unfavorable. In the case of the amino acid proline, which has this cyclic structure here, it's able to adopt either the trans or the cis conformation. And this is extremely useful in processes such as protein folding or conformational switching between protein states. There are many ways that we can study protein structures and conformational exchange, but in this case, NMR provides some unique advantages. The first advantage is that we're able to study proteins in the solution state, and this is where they normally exist, in our body fluids, in the cytoplasm. So we can actually tailor our NMR experiments to physiological conditions, or we can push them to the extremes and look at the proteins under the unfolded state conditions. We can also look at dynamics on a range of timescales, like I said before, though for NMR, we're really focusing on timescales from the picosecond to seconds. And we're also able to study structures in conformational exchange with atomic resolution. So that brings me to my research goal, which was to study the conformational plasticity of proteins using solution NMR. I'm going to talk about two of the projects that I've worked on. The first, which is kind of my baby to be honest, is the characterization of the C-terminal domain of the hemolysin 2 toxin. And I also worked on another project looking at the structural changes as GCN4P, which is a coiled-coil protein, is going from neutral to acidic conditions. So, like any good story, I'll start at the beginning with what I call the mutant model. But first, I want to give a little bit of an introduction on pore-forming toxins. Pore-forming toxins are characterized into two groups, and they're based on the secondary structural element, whether it's an alpha helix or a beta sheet, that forms the transmembrane portion of the pore. In the case of beta pore-forming toxins, they're comprised of mainly beta sheet character, which are these strands that you see. They lack a hydrophobic stretch in the primary amino acid sequence, and because of this, they need to assemble into higher order oligomeric states in order to form the transmembrane pore. Their mechanism of action is that the toxins are released as water-soluble monomers from the bacteria, and they bind to a surface moiety on the host cell. This allows them to concentrate and oligomerize into this pre-pore complex that undergoes a conformational change to a lytic pore, which kills the host cell, but it's also providing nutrients for the bacteria. Hemolysin 2 is a beta pore-forming toxin that comes from Bacillus cereus, which is a soil-dwelling microbe that's found to be pathogenic. And originally it had its one-hit wonder with food poisoning, but then it started becoming implicated in a lot of other infections like septicemia. So when stuff starts to kill people, we take notice of it. Hemolysin 2's closest relative is alpha hemolysin, which shares a 33% sequence identity. But hemolysin 2 has a C-terminal domain that has a unique amino acid sequence and no known homologs. Some of the things that we do know from literature are that HLY2C, which is how we'll be referring to the C-terminal domain for the remainder of the talk, it doesn't inherently show pore-forming activity by itself. And the pore can form without HLY2C, however, hemolysin 2 isn't as toxic without the C-terminal domain attached. And we think that it might either be involved in oligomerization or some kind of stabilization of the pore complex. But in order to start solving that mystery, we need to know what it looks like. So that became my job. But as any good structural biologist, I am here to entertain you with pretty rainbow cartoons. Shown here is a 2D NMR experiment that's basic in nature, but it tells us a lot of information, and this is the 2D HSQC. And what this is showing, and I'll explain this because it's going to come up a couple of times throughout the talk, is the proton and amide correlation in the peptide backbone. So for every amino acid in the peptide sequence, we should expect one peak in the HSQC spectrum. For the wild type spectrum, the HSQC that's shown here, there should be 94 peaks, but before you start counting, there's more than that, and a lot of them show this doubling. And this was found to be due to cis-trans isomerization of the sole proline at position 405. Now, when that proline hinge is mutated out and replaced with a methionine, that peak splitting goes away, and we're left with one ambiguous data set that is trans-stabilized. So we're only looking at the transform here, and the cis goes away, which leaves me as a novice grad student a much easier time to solve the structure with, and also less ambiguous assignments. The way that I'm able to solve NMR structures is kind of how I think of it, like a 3D puzzle. And the restraints are the different types of puzzle pieces. So one of the major factors that we need are distance constraints, which we're able to obtain from spectra that are called nosy spectra, which has been the butt of many jokes because picking nosies has been my life for the past like 5 years. What these show are interproton distances between 1.8 and 5 angstroms, and that gives us information on local structures. Hydrogen bonds, which were obtained from a long-range HECR experiment, were used to constrain secondary structural elements such as alpha helices and beta sheets, and dihedral angles limit the torsion of the backbone. All of that can be put into your favorite structure calculation software, and after months of careful pruning and picking, you get this. Not exactly the best thing ever. I called it spaghetti and helices for a little while, because this is really what it looked like. So basically what I have to do is I have to go back and I have to compare the results of my structure calculations to the data that I've obtained from NMR experiments and look at the differences between them and basically prune the violations, making updated assignments or increasing bounds, and that can all be put back into the structure calculation software. And we go through this iterative process, which was challenging at first because Hemolysin 2C doesn't show, it has no homologs to go on, so I don't have like a model structure to figure out what this thing looks like. So it took a lot of combing through NOEs to try and figure out what this was, but eventually there was a network of NOEs that appeared that held the structure together. And what was interesting was because I didn't have a model, I actually used a paper model that I made myself because it put things in a perspective. I knew the hydrogen bonding patterns based on the experiments that I had, so I knew that there was a beta hairpin here and a beta sheet here, but I was trying to figure out how they came together. And eventually the NOE network appeared, and I was able to get the pretty rainbow cartoon I'm about to show you spinning around. This is the structure of HLY2C, and it's split into two subdomains. It's got an N-terminal alpha helix, followed by a beta hairpin of strands 1 and 2, and then on the other side of the structure is a non-consecutive beta sheet of strands 3, 4, and 5 with alpha helix 2 that sits between strands 4 and 5. And this is a novel fold. This is a new protein topology that was discovered in our lab. The structure comes together in a pseudo-barrel type fold, and it has 3 hydrophobic layers. So the NOE data that's shown here illustrates the data that's shown here, where the way that this is put together is that the aromatic residues, or those ringed residues that are coming from the second half of the molecule, are interacting with hydrophobic side chains that are jutting inwards from the alpha helices that flank the structure on either side. Now P405M doesn't just have one conformation. It was found to form a dimer. This was initially discovered by Dr. Kaus in Olson Lab at Wesleyan University when she was purifying the protein for us initially. And one of the steps is gel filtration chromatography, which separates molecules based on size. And she found in the elution profile that there was a peak that corresponded to a dimer. This was also visualized in NMR experiments, where these minor peaks that are due to the dimer are shown by these little pink X's here. So this also indicates, because of the peak dispersion, that this is a folded dimer. And this dimer shows up at higher temperatures and concentrations. So just taking this amino acid as an example, where the dimer peak results on top and the monomer peak is on the bottom, we can see that the dimer is favored at higher temperatures and concentrations as opposed to lower temperatures and concentrations. There's a couple ways that we were able to confirm this using NMR. The first was T2 relaxation, where we were able to compare the T2 relaxation rates of the monomer and dimer. Larger molecules tend to tumble slower in solution, and so they have shorter T2 relaxation times or faster T2 relaxation rates. And this was shown in the data, where the dimer has a rate of 4.77 per second, while the monomer only has a rate of 10.18 per second. So the dimer relaxes more quickly. Another way that we're able to do this is using diffusion spectroscopy, or DOSI. And this looks at the translational diffusion of the protein in the tube. So there's an initial 90-degree pulse, which just excites the nuclei. Sorry, there's a lot of NMR. But they're left to process, and then there's a pulse field gradient that gets applied that dephases the spin. So basically what that leads to is a loss of signal. For proteins that don't move as much, a refocusing pulse is applied, and those spins get refocused, leading to the return of signals. We're able to see them again. For molecules that diffuse quickly through the tube and are not in the same place that they were before, those spins remain dephased, and that leads to a loss of signal. So we don't see those anymore. And based on the self-diffusion coefficients, we can determine which molecule is larger. So as expected, the dimer has a slower rate of diffusion than the monomer. And there was also a nice little check to this, where we were able to look at the radius of hydration, and this is just based on the amino acid sequence, so it's kind of a check. Where the radius of hydration is defined as a sphere that's going to have the same diffusion rate as the protein, given the number of amino acids. So if we plug in the numbers for the monomer and dimer to n, which is the number of amino acids, we get that there's going to be about a 1.2x difference between the two. And that is what we're seeing, which is kind of nice when science works. Okay, so next step, I need to assign the peaks in the HSQC. I was able to do this using a series of 3D experiments that are through Bond, the HNCACB, the HNCO, and a TOXI experiment. The HNCB and HNCO give me backbone correlations, while the TOXI gives me more about side chain correlations. But to actually solve the structure, I need more information than that. There are edited nosy experiments, this is what we typically use, that show interproton distances between labeled chains. But there's also a 13C filtered nosy experiment that I'll explain in a moment, as well as using a paramagnetic probe that I'll also explain in a moment. To determine the dimer interface, I used a neutrally charged paramagnetic molecule called Temple. It's relatively small. Paramagnet means that it has one unpaired electron. And what that means, it's going to align strongly to the external magnetic field, and it's going to make the nuclei that are around it relax quickly. So that leads to a loss of signal intensity. So basically, peaks that disappear are those which are in direct contact or solvent exposed with Temple. Those which broaden out or show like some peak attenuation are in transient access to Temple, so maybe some dynamic regions. And the peaks that disappear, the peaks that are kept are those which are supposedly in the dimer interface, or at least that's what I was hoping for. And when I look at the well-resolved peaks for the monomer and dimer, I find that a lot of the protected residues are coming from the second half of the molecule. So my expectation is that this beta sheet is somehow involved in the dimer interface. C13 filtered nosy experiments were done on a mixed labeled sample. So for NMR, in order to be visible, we need isotropic labeling. For chains that are not labeled, we don't see them as readily. And the C13 filtered experiment looks at intermolecular proton interactions where resonances are selected based on protons that are transferring magnetization from a labeled chain to an unlabeled chain and back for detection. So what happens to the intrachain ones that are labeled? We don't end up seeing those because purge pulses will knock them out. So interestingly enough, based on the C13 filtered nosy experiments, there were interactions that were happening between hydrophobic residues of beta strand 3 and beta strand 5, which are illustrated here. So one of the more distinct NOEs was from I412 to this tryptophan 372, and there were also interactions between K410 and I374. So a network was starting to form, and this is what I started to go on. So indeed, the dimer interface is in the second half of the beta sheet. But what I'm starting to see from structure calculations is that there are two populations of structures that I have, what I call the compact model and what I call the extended model. And this is likely due to a lack of restraint, holding it like this, or maybe it is like this, but that needs further determination. So the next steps for the dimer project are to determine the KD from using data from a dilution series, to determine the thermodynamic parameters from a temperature series that I've done, and also to better characterize the dimer structure, which is what I'm working on. So now that I know what HLY2C looks like, I can use that as a model to solve the structures of the wild type cis and transforms, which are shown here. They look quite similar to one another, but if I superimpose them on top of one another, there are definitive differences between them, and it looks like mainly the changes are focused around where this proline loop is. but that analysis doesn't really get you publications in Nature, so I have to quantify that. Shown here are the superpositions of the cis and trans structure bundles that were solved. There are 25 structures each, and not only does this show the structural precision, it also shows the differences that are happening between them. And this graph is also exemplifying that. So for the blue and orange lines are the differences within the cis and trans ensembles inherently to those ensembles. The black line is comparing the cis and trans forms to one another. So anywhere where the black line is larger than the blue and orange lines are basically the locations where the most structural change is occurring due to proline isomerization. So the largest change is happening quite clearly in the proline loop. And that extends to shifting the alpha helix 2 over here. There's also change happening at the top of the beta hairpin in the first part of the molecule, which you can also see on this side here, where the laser pointer is. But there are also changes that are happening at the bottom of this structure, at the end of alpha helix 1, and the loop that's adjacent to it. And that was found to be due to hydrophobic, or movement that was being propagated through the hydrophobic core to the other side of the structure. But rather than think of this as a static thing, they're not two structures independently of one another, they're one undergoing conformational exchange. And that's shown here where the cis loop is up and the trans loop was where it comes down. So in a 2006 paper, they showed, Alexander Solinen's group showed that the pore structure tended to change with temperature. And so something I wanted to see was, does HLY2C equilibrium also change with temperature? So I did a titration series using NMR, and this is just an example of one residue that's changing, where it appears that trans is favored at higher temperatures while cis is favored at lower temperatures. Using this information, I was able to generate Van Hoff plots, which look at the change in the equilibrium as a function of temperature, going from cis to trans. And using the Van Hoff equation, I was able to extract the thermodynamic parameters, change in enthalpy from the slope and the change in entropy from the y-intercept. And averaging those over the structure, over the peaks that were well resolved, I was able to deduce that trans is entropically favored while cis appears to be enthalpically favored. Another thing that I wanted to look at was if there were any differences in the electrostatic landscape as cis changes to trans or vice versa, because maybe this could open up a binding interface, or maybe it would provide more of a result as to, say, maybe there's a binding partner. So I looked at each of the 25 structures for each ensemble, and I didn't see any major differences between the two. The molecule is overall pretty positively charged. However, there are some lysines that are concentrated on the bottom of the molecule, meaning that there are more positive charges on the bottom. And from a homology modeling experiment where we use alpha hemolysin as the pore structure, just because hemolysin 2 doesn't have a pore structure that's been solved yet, we were able to tag hemolysin 2 onto the end of that. And one of the things that we thought is that the lysins at the bottom of the structure might be interacting with the cell surface somehow. So to summarize this part of the talk and draw some conclusions from it, I did solve the structure of multiple conformations of HLY2C using solution NMR. P405M revealed a novel fold, which was cool, and that was used as a model to determine the structures of the wild-type HLY2C. The structural differences between cis and trans are mainly centered around the proline loop, though there are differences that are propagated throughout the structure due to the hydrophobic interactions in the core. And P405M might provide some insight into the transform of the domain. Some of the possibilities involved might be that HLY2C is either helping in oligomerization, or it might be used to stabilize the pore complex somehow. And the function of HLY2C is still unknown. So, if anybody needs a thesis project. And further characterization of the dimer is also necessary. Another project that I worked on with the collaboration of a wonderful undergrad that was in our lab, Megan Brady, I've also worked on, where the research question was looking at the role of the ion pair contributions to the stability of the GCN4P coil-to-coil protein. And it also takes a look at the structural changes that are happening as the protein goes from a neutral pH down to an acidic pH, where we're titrating the acidic side chains. GCN4P is a parallel homodimer that has a motif called a heptad repeat, which is just a repeating series of 7 amino acids that are denoted by letters A through G. And on this helical wheel diagram, at positions A and D are where hydrophobic side chains generally reside, and then at positions E and G are generally oppositely charged amino acid side chains that form interhelical salt bridges that are thought to stabilize the structure. But what happens when these salt bridges get broken? To better investigate that question, NMR structures will solve at 3 different pH values. pH 6.6, where acidic side chains have negative charge. pH 4.4, where they're half charged, as that's around the pKa of the acidic side chains. And then pH 1.5, where everything is neutralized by being protonated. Shown here are the 20 lowest energy structures, and they show good agreement within the bundle to each other. But one thing to notice is that when compared to a crystal structure to ZTA, the RMSD goes up as the pH goes down. So these are changing from the crystal structure. To determine whether these salt bridges were the things that were affected, I actually measured the distances between the charged side chains. And the average values for all of those are shown at the bottom, where the differences between K15 and E20 and E27 to E22 are increasing as the pH decreases. So these interhelical salt bridges are being broken as the pH is decreased, which is what we expected to see. To investigate what's happening with the superhelical structure, we can look at what are called Crick parameters. And these were proposed by Francis Crick in 1953, and it's basically a breakdown of the coil-to-coil into these parameters that show how coily the protein actually is. Where the R0 is the superhelical radius, so going from the center of one helix to the center between the helices. The R1 is the helical radius, so that's just for one helix on its own. The superhelical frequency is basically the twist of the overall superhelix, which I'll talk about in a little bit, while the omega-1 is the just helical frequency. The D is the rise for residue, and Z is just a chain offset from the axis. So analyzing these Crick parameters over the average of the structures, we found that R0, which is the superhelical radius, is increasing as the pH decreases. So basically, it's becoming less superhelical as we decrease the pH. In the ideal, so these chains are moving apart, I'm sorry. The R1, however, which is the helical radius, so just the radius of one alpha helix, is not really changing that much. So the alpha helices by themselves are staying kind of the same. The W0, which is the superhelical frequency, which is basically how twisty the actual superhelix is, increases. So in an ideal helix, the measure of this is degrees per residue. There's negative 3.6 degrees per residue, with a negative indicating that it's just a left-handed helix. So we get a full 360-degree turn over 100 residues. At pH 6.6, if we compare these, we have negative 4.78. So it takes 75 residues per turn to create the superhelical structure at pH 6.6. If we do this for 4.4 and pH 1.5, we get that this number starts to increase, meaning it takes more amino acid residues, or it takes more amino acids to actually form the superhelix at all. So it's a much looser structure is basically what that turns into. For the alpha helices themselves, that's not changing much. So again, the alpha helical structure, like the monomeric form, is preserved. And the rise and the offset don't change much. To further validate the unwinding of the superhelix, we use a cross-validation of the pH 6.6 and 1.5 structural restraints. where structures were calculated using simulated restraint sets, where we basically just took out different residues depending on their distance from one another in the sequence. So for example, the difference between I and J, which are the two residues, are how far apart they are on opposite chains. So for example, in the K15 to E20 interhelical salt bridge, they are 5 apart from 15 to 20 on the opposite chain. Oops, sorry. So removal of long-range restraints, we see that there is a decrease in superhelical structure, but it's still maintained somehow with more long-range restraints removed, or with more restraints removed, pretty much all the restraints removed at that point, except for short range, the superhelical frequency still goes down. So again, we're loosening the helix here. When only the short range restraints for pH 6 are removed, the superhelical structure is maintained. So really what's holding this together are the long range restraints that are coming together to make the supercoil more coily. For pH 1.5, with the full data set, the superhelical frequency is still not there. And despite putting back, despite putting in the long range distances that are lost from pH 6.6, so the interhelical salt bridges, this does increase the superhelical frequency, but it's not quite as much as the pH 6.6, meaning that these are distinct structures and it's not just due to a loss of NOEs or just lack of precision. So the conclusions are that the superhelical structure of GCN4 unwinds as pH is lowered, and this is due to the loss of interhelical bridges, and each chain of the dimer is maintaining its own alpha helical structure. And that's nicely seen by this transition. So my research goal was to look at the conformational plasticity of proteins, and I've solved a lot of different conformations using solution NMR. So I have reached my goal, I guess. I want to thank everybody for coming. I want to especially acknowledge Andre for having me in his lab for the last 5 years and tolerating me and pulling me out of all sorts of rabbit holes. So thanks for that. Thank you for the project. Thank you to Dr. Olson for also giving me this project. I've really enjoyed working on it, and thank you for the time I was able to spend in your lab learning ITC and protein purification techniques. Dr. Robinson, I was talking to you at the beginning. Thank you so much for all of your help and all of your wonderful insights. Dr. Alder, same to you. Thank you for all of your insights on the project and all of the questions that you've asked your guys. And thank you to Dr. Majewski for helping me with not only NMR, but also with a lot of computational issues that cropped up with structure calculations. Our collaborators at Wesleyan, Dr. Kaus, Teschke Lab Group as well, shout out to them because you guys have sat through many a presentation and given me feedback, and I sincerely appreciate all of the suggestions that you've made. My lab members, Becca and Richard, you've been there. Also to past lab members who have also been extremely helpful. Christina Harprecht, Helen Bellato, and Megan Brady, who I collaborated with on the GCN4 project, and I was lucky enough to help with structure determination. And obviously all of my wonderful friends and family who've tolerated me being a hermit for the last month. And Marcos, you have been a trooper through this. You haven't had a wife for like the past at least 3 months, so I'm really sorry. But hopefully you get me back. And I don't know. Oh, sorry. Yeah, thank you everyone for being here.
Okay, so the bell went off, so I guess it's time for general questions from the general audience. And friends and family can ask them as well. You can turn up the lights a little bit so we can see.
Yeah, sorry. Yes?
So in the HLY2C, you speculate that the positive charges near could be interacting with the cell surface.
Yeah.
Is there any thought of collaborating with anybody to do some mutants and actually test in Bacillus if that occurs?
That would be really neat to do, yeah.
Yeah, one of the world Bacillus experts here at UPenn. Pete Zettler.
That's true. I think we had reached out to him previously about this. It's definitely worth looking into. It's difficult to purify this domain, not the domain, but it's difficult to purify the toxin itself, but to look at it in the context of Bacillus, that would be an interesting approach. As far as the positive charge, I mean, yeah, we could, it's difficult to try and pin what it is exactly it might be binding to, so to look for a specific binding partner was something that I kind of had to give up on myself originally, just because it could be anything at this point. But yeah, it does merit a collaboration, be interesting.
Oh, Richard.
So in your structure of the dimer, where you have it sort of closed and open, do you think that that's just the number of restraints that you were able to collect for the dimer, or that it's actually in those two conformations?
I think it might be a restraint problem, just because there's not a whole lot of NOEs that are holding it together. I also pulled some out because I wanted to see what would happen if there were like key residues that were involved, but so far I want to keep those, I want to keep like certain key interactions and kind of see if the data supports one conformation or the other without actually biasing it.
Sure.
So it's just testing new restraints being put in from the nosy at this point.
Any other questions? All right, so if there's no further questions, we'd like to, did you have a question, Charlie? Okay. If there's no further questions, I'd like to excuse the general audience, and the committee members will now participate in the exam, which is going to be on the second floor. So we're moving back up to the second floor.
Thank you.